Thank you so much, Bhavana. And it's really a pleasure to come here again. I've been here 2 weeks ago, 3 weeks ago. At that time, I actually was connected from Brazil, where I am from. I'm ophthalmologist as a background, surgeon, mainly on the theater segment, and I've been working in gene therapy most of my business life. And I got passionate about gene therapy and what we can propose as therapies to bring to patients. Before joining AAVantgarde, I had the pleasure to work with Novartis in the rare disease team. And then our team was responsible to bring Luxturna for RP65 inherited retinal dystrophies. And then I was leading the medical affairs team outside the United States. So a lot of collaboration with our Spark team from the US. Now it's part of the Roche team. And then also under Novartis, rare disease, I have the pleasure to work with the neuroscience team and then to launch Zolgensma, which is a medication for spinal muscle atrophy. It's a gene therapy as well. It's not intraocular treatment, but it was intravenous. And then we had the opportunity to have a lot of engagement with the community from spinal muscle atrophy. So it's really, really exciting. Joined AAVantgarde about a year ago. So I'm leading the medical affairs organization from AAVantgarde. And then we are really proud to be with here. And I just mentioned to Bhavana, let's keep this contact. Let's keep the connection with the Stargardt community. And then let's plan something for the next few quarters in 2026. So let me share my screen. Let's make sure that I have good connection in Zoom. And I need to share my screen first. This one here. Okay. And Bhavna, you should hear my sound now. I hope it's okay.
Yeah, I can hear it very clearly. Thank you.
Perfect. Perfect. Thank you so much. Okay, so AAVantgarde. We are a biotech in the clinical stage, and our focus is to deliver large genes into the retina cells. We are focused on inherited retinal dystrophies, and then we have two programs which are at a clinical stage. The Stargardt disease with ABCA4 gene, and then retinitis pigmentosa secondary to Usher1B. That's caused by allelic mutations of the Bio7A gene. At AAVantgarde, we develop transformative gene therapies by gene augmentation for patients with rare disease. And then our dual AAV platform is designed to deliver large genes. I'll get into the discussion in a few slides. What does it mean, a large gene or a small gene? But what it's important here is that we are trying to address pathologies that, because of the size of the gene, would not be able to fit into a standard AAV pathway, similar to the example that I've mentioned to you with Vortigern and Parvovec for RP65 variations. So we have two programs which are on a clinical stage. I'll have the chance to share a little bit more details about our two studies that we are conducting at AAVantgarde on Stargardt disease. In the previous meeting, we had the chance to go a little bit more on the genetics of the Stargardt disease, but it's an inherited retinal dystrophy. It's a rare disease, and it's caused by variances of the ABCA4 gene. Stargardt disease, it's affecting the back of the eye. I have one slide to explain that. And then it's affecting the central vision. The patient would start to see a blurry spot in the center of the sight, which may progress in time and obscure, and then to reduce, be the cause for the reduction of the visual acuity of the patient. And that it will make difficult for the patient to read, to recognize faces, and to see fine details. This is the eye, and then I went in detail in the previous meeting, but I'm ophthalmologist, I'm biased. I believe that the eye, it's a fantastic organ which has many functions. But if I would need to summarize the main function of the eye, I would say that the eye, it's important to take the light, which is coming from the outside, go through all the layers of the eye, and then to touch the specifically point on the retina of the patient, in which we have these important cells, rods and cones, which have the ability to transform the light coming from the outside into neuronal impulse. And then these red and cone cells, they're connected to the visual cortex of the patient so the patient can see. So let's get a little bit more in detail about this connection here between these two important cells of the retina. So in the left here, we have a photoreceptor, which is this top cell here on the top, which is connected to the pigmented retina epithelium. So this is a very important connection. And the photoreceptor is the cell which has the ability to transform light into neuronal impulse. What I want to describe here without getting too much into details is that the function of the ABCA4 gene, it's very important in this segment of the outer segment of the photoreceptor, in which the ABCA4 protein will position proteins in order to be absorbed or phagocytized by the cell which is down here, the retina pigmented epithelium. So if all goes well, all this function is working very nicely and properly. There is the transformation from the light coming from the outside into neuronal impulse. And then as a consequence of this metabolism, we have very, very little accumulation of these green dots here in this cell in the bottom. Those green dots, they're called lipofuscin granules. And then what happens on a cell with a defected ABCA4 gene, which is the case of the Stargardt disease? The ABCA4 gene is not functioning properly. It's not providing all the function and the benefit and the uh, the function that it's expected to do. And then what we can see here is that we have a accumulation of a much larger number of these green dots, which are lipofuscin granules, and this is not good for the eye. So we want to avoid or to reduce here. And then what we can see here is that you have a nicely cell is like a squared shape, and then the shape of this cell is not healthy anymore. So it's accumulating lipofuscin, which it's far beyond what the cell could absorb. So what we're trying to do with any treatment with Stargardt and trying to normalize as much as possible this process. So we have much less accumulation of lipofuscin granules inside of the retina cells. So when we get into a discussion about gene therapy, we talk about vectors. And for us, there are different vectors in medicine. A vector is a way to bring a medication into the patient. It could be a pill, it could be an eye drop, but in our case, we need to have a specifically transporter, someone to transport the DNA of interest for us, which is the ABCA4 gene inside of our host cell. I'll just do a very short analogy with you go to a shop, you buy a big TV screen, and then you need to transport this television to your house. So it's a transportation of an element. If we bring this into gene therapy and to vectors, what does it mean that in this case here, our truck is the viral vector, and then the big box with the TV screen, it's our ABCA4 gene. And then we want to deliver this inside of the retina cells by doing a subretinal injection. So we have different vectors, viral vectors, which can be used. Viral vectors is one of the most used ways to deliver a gene in the eye. We have adeno-associated virus. we have adenovirus, and then we have also lentivirus. Those elements, they would vary on the size of the capacity, which means what is the size of the DNA that they can carry and transport in this system. And then it will also vary on the ability of this virus to transfect the host cell, and then what is the immunogenicity that will bring into after this treatment. So one of the very most common is the adeno-associated virus. So that's what we use. That's what we use for the AAVB039, which is the medication that we are investigating at AAVantgarde for Stargardt disease. And then we start with AAV vector. We remove the content of this virus, and then inside we introduce the gene of interest for us. So for us, it's a ABCA4. So our DNA is packed inside of the AAV cell, and then by injecting billions of units of this virus inside of the retina, it will be a subretinal injection. We inject billions of units of this virus, and then it will transduce, or it will penetrate the host cell. Remember, what is the virus? Think about the virus of the flu that we have, very common during winter time. The virus, it will penetrate, and it will penetrate inside of the host tissue and deliver its own DNA. But what we have done here, we are using that same process. We are using that ability of the virus to penetrate other cells, but we actually removing the DNA of the normal virus, and we replacing that of our DNA of interest. And for us, it's a ABCA4. What does it mean, a large gene? Think about this truck here in the very beginning, that the truck can only fit a mid-side screen, for example. And then, but if I would need to have a larger screen, I would need to split into two pieces and use two different trucks to deliver this equipment into our final destination, which is the retina cell. So what we are doing here, in my example here that I've mentioned with ORP65, which is Vortigen and Pavlovec, it's another disease, it's inherited retinal dystrophy. That gene is small. It's small enough to fit in one AAV vector. In our case, for ABCA4, the size of the DNA is larger. So what do? We split into two pieces. We introduce this into separate AAV vectors, and we inject same quantity, same volume inside of the eye, and then those two pieces will penetrate the same retina cell, and they will connect to each other. And then the process to the production of the protein, which is important for the function of the eye, it will happen as a normal procedure. So now I have a video here, and you should listen sound.
Many patients live with inherited retinal diseases, yet vision impairment often goes unseen. The human eye is a marvel of biological engineering, where rods and cones convert light into electrical signals, enabling us to perceive the world in colour, depth and motion. IRDs remain an unmet medical need, as photoreceptors gradually degenerate, leading to progressive vision loss and underscoring the urgency for new therapies. Adeno-associated virus, AAV vectors, with their capacity to encapsulate a therapeutic DNA of 5 kilobase in length, offers hope. AAV vectors can be administered via subretinal injection, an intraocular procedure that delivers vectors into the virtual space between the retinal pigment epithelium and photoreceptors, creating a localized and reversible bleb. However, many IRDs stem from mutations in large genes that exceed AAV's 5 kilobase cargo limit. To overcome the 5 kilobase limitation, AAVantgarde has developed two innovative platforms, dual hybrid and intine protein transsplicing. Both approaches split the gene into two halves, packaging each half into separate AAV vectors. Inside the cell, they reassemble to produce a full-length protein. The dual hybrid strategy relies on a recombination event between both DNA molecules carried by the AAV vectors inside the nucleus of the same photoreceptor cell. Once the DNA is transcribed into mRNA, splicing removes the remaining sequence between the halves. The ribosome then reads the mRNA, producing a functional protein. Intine protein transsplicing follows a similar strategy. However, instead of recombining DNA, each AAV expresses an mRNA molecule that is translated into different protein fragments. Thanks to the intine sequences, the two protein fragments self-assemble, forming a complete and functional protein, while the intine fragment is excised. With these advances, there is potential to restore photoreceptor function and vision. AAVantgarde is paving the way for transformative gene therapies, bringing hope to those affected by IRDs.
So let me just recap very shortly what was the mechanism of action that we've just heard here. It might be a little bit complex, but remember the vector here, it's the truck, and then the DNA that it's inside, it's our sound equipment, for example, and then the house that we have for us, it's our retina cells. So we start by using two different vectors. This is in the top here, it's the adeno-associated virus, or AAV. We remove their viral content, we split the ABCA4 gene into two pieces, we include it inside, and then we inject equal volumes under the retina of the patient, so subretinal. When you have the production of the half proteins, in these cases here, those two little pieces here, they're called inteins. Think about like metabolic magnets. They will find each other inside of the cell, they will connect, and they will produce a full-length protein as if the body would do in a normal cell. And then we have this excised intein, which is metabolized. But what is important for us, at the end of the process, we have a full-length protein of ABCA4, which is functioning as a normal cell. So what is the clinical development program that AAVantgarde is bringing to the Stargardt disease? We have two ongoing studies. The first study, it's called STELLA. We do have an Italian origin, and we're very proud of that, so that's why we call it STELLA and CELESTE. STELLA is our natural history study. It's a study which is ongoing. We have sites open in the UK, in Europe, and then in the United States. And then what we're planning to do is to follow patients without treatment, but with a very structured way to follow as we would do for a interventional trial. We're going to follow these patients and to observe how do they perform or react, how would the disease progress or not in 12 months period. And this is a 2-year follow-up study. We are including patients between the 8 and 55 with a genetic variants on the ABCA4 gene. Once these patients, they would integrate for 12 months, we would enroll them on the interventional trial, which is the CELESTE study. This is a 5-year follow-up study. there are two parts of this interventional trial. The first part we call the dose ascending study. There will be 3 cohorts because we're going to investigate 3 doses of the AAVB 039 in this group of patients. This group of patients will be part of our safety assessment and then dose assessment. Once we have defined that the drug is safe to move forward, and then we're going to define what is the final dose of this medication, we would get into what we call an expansion cohort for adult and pediatric patients. The follow-up of this trial is about 5 years. And then in the UK, their sites already open. We have the Moorfields Eye Hospital and the Retina Clinic in London, both sites in London in the UK. We have sites open in the United States, and then we're about to open sites in Europe as well. So when we do the assessment of this, both of the studies, we're trying to associate what are the signs and symptoms that the patient would refer. So for example, here on the top of the screen, you have the changes on the sign of the patient with this blurry vision that starts in the center of the vision and it's progressing. And then we want to follow up this with a number of visual assessments. So I'll just list some of those. Ocular coherence tomography, or OCT. We want to do a screen. It's almost like an MRI that we do for abdominal or lung pathologies, for example. But we have the ability to do a very nice scan and to see all the different layers of the retina and to follow up the progression of the disease and to see any signs of effectiveness of the medication. We have another measurement which is called fundus autofluorescence. It's this measurement here in the center. And then, as you can see here, the anatomical change that we see when a patient will refer, starting to see some blurred vision, is that on this central alteration of the on the macula area on the retina of the patient, and then this is typically increasing in size, and then that was the reason that it's causing this blurred vision to the patient. We have other assessment, which is called microperimetry, which is assessing in each of these specifically points in the back of the retina, what is the lowest level of light that the patient can perceive in that specific dot of the retina. So it's very, very precise. And then, of course, one of the most common assessment in ophthalmology is best corrected visual acuity. This is when the patient is reading letters, and then we are counting how many letters that patient can read in that visit. In addition to that, we have a number of patient questionnaires that are specifically designed for low vision, and they're specifically designed for pathologies which are assessing inherited retinal dystrophies. So with all this information, we want to first understand in our STELLA trial how it does disease progressing over 12-month period. And then when we would dose the patients in the CELESTE interventional trial, we'll try to understand what is the change that the medication might be bringing to the eyes of the patient. So in few words, if you go now with CELESTE trial, which is our interventional study. Remember, we have one part which is the dose ascending piece with 3 doses, and then we move into a expansion cohort. All clinical trials start with an eligibility assessment. We're going to do a lot of the measurements that I've just mentioned in the previous slide, and we'll also do a genetic molecular confirmation that the patient do have biallelic variances on the ABCA4. This is very important because there are many other diseases that could mimic a Stargardt disease, and only a confirmed genetic molecule examination can confirm that it's a Stargardt disease, genetically speaking. Then we'll do a baseline assessment with a lot of the measurements and many more that I've just mentioned in the previous slide. The surgery date, that's when the day 0 of the clinical trial would start. That's where we would have a surgical injection of our medication, which is AAVB 039. And then it might sound a lot, but we're literally injecting few drops. It's a very small tissue, it's a very small region. But we want this subretinal treatment be as precise as possible. So there's a lot of training, there's a lot of discussion with the surgical team to make sure that we all know how to best perform for the benefit and safety for the patients in that day. Then we'll get into a 5-year follow-up of this trial. And here, this is the end of my talk. We're really proud to be able to bring this investigation of AVB 039 to Stargardt patients. The study is in development program. We're going to work here. We're going to communicate back to this community as we're progressing with the studies. But I would like here to give it available for you, our email contact on an AAVantgarde's website where more information is available for patient. So at this point, Bafana, I'll just stop the mic, and then I'll just stop sharing so I can open for questions from the group. Thank you.
Perfect. Thank you so much, Dr. Spero. It's so interesting, such an interesting presentation. I'm just going to stop recording.